All right, everyone. Good afternoon. Hello to all of you, wherever you are. Welcome to this new edition of the Worldwide Neurodevelopment Seminar Series. Our guest today, our presenter today is James Briscoe from the Crick Institute. Hi, James.
Hi, nice to see you.
Nice to see you too. Nice to be live with you and with all of you. Before I tell you a little bit about James' background and let him have the front stage, I'd like to remind the most junior people in the audience, in particular PhDs, senior PhDs, or postdocs, that we're trying to implement a new feature where before the main speaker, there would be a 3-minute short presentation, poster style. I haven't received that many applications. So if you're interested, don't hesitate to hop in. If you know people who are interested, don't hesitate to advertise. I put a Twitter ad related to this. So, just hop in. Even if you're not feeling comfortable with your presentation skills, the idea is to help you get there. There'll be some, training sessions. So if you have a good science to share wherever you live, and especially if you live in a place where you're not necessarily super exposed to your peers, don't hesitate to send an email. I'll send the link on the sidebar. All right. So this being said, let's get back to the main hero of the day. So James did his bachelor's degree in microbiology and virology, a PhD in oncology, so a broad cellular biology training. Did his postdoc with Tom Jessel at Columbia University, where he developed, I guess the word is well chosen, developed his interest in spinal cord and spinal cord development. Has moved to the MRC in 2000, and I'm not seeing on my list here. When did you join when did you move into the Cricks, James?
The Crick started in 2015, I think.
2015, yes.
Yeah.
2015. And in 2018, he became editor-in-chief of the journal Development, which is a really nice venue for I guess all of us interested in development in general and in neuroscience in particular. So I'm going to stop here. He will be telling us today about his main field of interest, spinal cord development, and the temporal controls over the emergence of this structure. James, the screen is yours.
Great, thanks very much. So let me see if I can work this. I'm going to share my screen. And hopefully now you can see my screen. So thanks very much for the opportunity. It's a pleasure to be able to present to you today. So, and get my pointer working. Yeah. So I'd like to tell you about some of our recent work in the lab over the last couple of years. But just before I get to that, let me just say a word about development. As Denise said, I'm an editor of Development, which, as you all know, is a premier journal in the field. And we're very keen to publish all of your neural development studies. But what I wanted to emphasize that in addition to being a journal, we're also a not-for-profit charity, and we support the field by giving fellowships, funding conferences and societies, and also initiatives such as a Node and Prelights. So remember, by publishing your next paper in development, not only will you gain the admiration and respect of all your friends and colleagues, you'll also be supporting your field. So please do get in touch if you would like to discuss a paper or indeed any aspect of the journal. I'm always happy to talk about development. So with that, let me move on to talk about some of our recent work. So I'm really a developmental biologist, and what I'm interested in as a developmental biologist is how do you make the right cells in the right place at the right time in the right numbers in a tissue. So part of the reason for this interest is because function follows form. The proper operation of an organ depends on having appropriate cell types correctly organized. And this is true whether we're thinking about the basic body plan of an individual, or specifying the digits on the ends of your limbs, or indeed organizing the neurons in the nervous system. And if we think about this last case, this is particularly relevant because there are hundreds of molecularly, physiologically, and functionally distinct neurons. These have to be generated and assembled into neural circuits. So what we'd like to understand is, how is this diversity of cell types generated, and what controls their arrangement in space and time? So what underpins that assembly of functional neural circuits? And to address these questions, we've really focused, almost exclusively on the spinal cord over the last 20, 25 years. As you know, the spinal cord is part of the central nervous system. It's essential for our ability to perceive and interact with our environment. And those circuits involved in perception and movement are spatially segregated in the adult spinal cord. So neurons located in the dorsal horns are mostly sensory. These receive input from the periphery and relay that information to various areas in the brain, or directly into the circuits necessary for locomotion and muscle control in the ventral spinal cord. So these locomotor circuits are comprised of various excitatory and inhibitory neurons. These control the activity of the muscle innervating motor neurons. And these are important for mediating the rhythmicity of muscle activity during breathing and walking, also coordinating muscle activity between the left and right sides of our body, ensuring antagonistic effects of motor neurons that innervate opposing flexion extensor muscles in our appendages aren't simultaneously activated. So understanding how these different neuronal subtypes are generated and form these circuits contribute to our understanding of the formation of the spinal cord, and I think is an interesting biological problem in its own right. But this has also served as a paradigm for other regions of the nervous system, and indeed for other tissues in general. So to understand this, we have to go back in time to the initial stages of neural tube spinal cord development. So initially, the spinal cord is composed of proliferating neural progenitors that form an epithelium, with the apical lining the future central canal, and basal being located at the lateral edges of the neural tube. So as postmitotic neurons differentiate from these proliferating progenitors, they migrate in a medial to lateral direction. And these early born neurons already display molecularly and physiologically distinct identities that depends on their dorsal ventral position. So ventral being close to your belly, dorsal, the skin of your back. Moreover, it's not only those newly born neurons which display distinct molecular identities. The progenitors themselves are also molecularly distinct. This is a consequence of gene expression which divides those progenitors into 13 distinct domains arrayed along the dorsal ventral axis. And each of these molecularly distinct domains generates a functionally identifiable neuronal class. So diversity and organization here is achieved by spatially segregating the generation of different cell types and by controlling the molecular identity of the progenitors from which neurons are produced. So here's what it really looks like. So you can see here is a set of gene expression, and these genes allow us to define distinct domains with the combination of gene expression at a particular dorsal ventral level, uniquely identifying that domain of progenitors. Moreover, we know from many years of work that manipulating this gene expression code within progenitors, so knocking out genes or misexpressing them, has predictable consequences on the neuronal subtypes generated from those progenitors. So this problem of neural tube patterning becomes one of understanding how you establish this pattern of gene expression within progenitors. So in ventral regions of the neural tube, this pattern is dependent on the activity of the secreted molecule sonic hedgehog emanating from the notochord underlying the neural tube, and this specialist group of cells, the floor plate, at the ventral midline. In the absence of sonic hedgehog, this pattern of gene expression is lost and this pattern of neurogenesis is aberrant. Moreover, a range of evidence suggests that sonic functions at long range in the ventral neural tube in a graded manner to establish this pattern of gene expression. So some of this evidence comes from these type of ex vivo experiments where a small region of the forming neural tube, the forming neural plate, is explanted from embryos and then cultured ex vivo in tissue culture. In the absence of any recombinant hedgehog, these explants adopt a dorsal identity here characterized by this blue gene, PAX7, expressed in the dorsal half of the neural tube. So addition of a moderate concentration, a low concentration of hedgehog protein, so 1 nanomolar, represses the dorsal gene and induces this red gene, OLIG2, characteristic of the middle of the ventral neural tube. At a higher concentration, so instead of 1 nanomolar using 4 nanomolar hedgehog, instead of that red gene, cells express this green gene, NKX22, characteristic of the ventral neural tube. So this looks like a classic kind of graded or morphogen response with gene expression dependent on different concentrations of the signal, and that concentration relating to the spatial relationships, the spatial location of gene expression in vivo. But of course, things are more complicated than this. So on the previous slide, I showed you the results at one fixed time point after 24 hours of exposure to recombinant hedgehog. Here I'm showing you one concentration of hedgehog, but over time. So that high concentration 4 nanomolar at 24 hours induces the high response gene, NKX22. But if we assay similar explants exposed for only 12 hours at high concentration, we see a transient expression of the low response gene, the red OLIG2 gene, before a transition of cells into the high response. And you can see this here if we count the number of cells expressing the low response gene. This initially increases, reaches a peak, and then decreases. And that peak is reached at the time when NKX22, the high response gene, is induced. So in order to predict gene expression, we need to know not only the amount of hedgehog protein, but also the duration of exposure. And that sequential of low and then high response genes is also seen in vivo. So we see this sort of gradual, these waves of gene expression where the low response gene is induced in the ventral neural tube, and then that transitions, that changes, cells change their gene expression ventrally to then induce the high response gene. So how does this work? So what I will argue is that to understand how cells are responding to this, the level and duration of hedgehog signaling, we need to understand how the genes themselves, the transcription factors that respond to hedgehog, how they're linked together in a gene regulatory network, and how that network defines the response. So this is the importance of this gene regulatory network in determining the response to hedgehog signaling. This first became apparent from classic developmental genetic experiments. So if we knock out one of those factors, one of those transcription factors expressed in progenitors, we can look at the consequences on the remaining genes. So here's an early example of this. So in the neural tube, you've already been introduced to a high response gene, NKX22, and that's expressed ventrally and mutually exclusively with another transcription factor, PAX6, expressed throughout the neural tube with the exception of NKX22 expressing progenitors. In mice lacking PAX6, PAX6 protein is obviously gone, but what's noticeable is this marked expansion of NKX22. So now the domain of NKX22 expression occupies a much larger proportion of the ventral neural tube. So what this indicates is that the genetics tells you is that PAX6 is a repressor of NKX22 and is responsible or contributes to setting the sensitivity of NKX22 to hedgehog signaling. If we remove PAX6, now there's a broader domain of NKX22, it's responding to lower levels of hedgehog signaling. So these type of experiments have allowed us to begin to reconstruct the regulatory interactions between the transcription factors in progenitors. And here's a little cartoon representing some of those results. As you can see, there's a lot of repressive interactions going on here, and particularly notice these sort of cross-repressive interactions between transcription factors expressed in adjacent domains of progenitors. And intuitively, you might think that these explain those defined switches. They explain how you transition from one identity to another. But I'd argue, I think just from looking at this network, it's difficult, and I'd say impossible, to really get a sense of how it works. So to try and do that, to try and get a better sense of what's going on here, one approach we've taken is to look at, to extract out smaller subcircuits from this larger network and examine those in great detail. I'm going to focus on, for the next few slides, on this circuit of 4 transcription factors. So 3 of these you've already met: PAX6, NKX2-2 and OLIG2. NKX2-2 and OLIG2 obviously dependent on Hedgehog signaling. The 4th one here, IRAK3. And the reason for choosing these 4 factors is they define 2 boundaries of gene expression. NKX2-2 is expressed ventrally in response to high levels of Hedgehog signaling. Its dorsal limb of expression corresponds to the ventral limb of OLIG2 and PAX6. And the dorsal limit of OLIG2 expression corresponds to the ventral limit of IRAK3 expression. And the kind of genetic experiments I've introduced to you suggest these regulatory relationships, these interactions between the 4 transcription factors. So we were interested in asking, can this circuit, can this transcriptional network explain this spatial pattern of gene expression? And if so, how is it operating? So one way we've approached this problem is to take a mathematical modeling route, a dynamical systems description of this. So take these cartoon representations of the circuit and convert them into a set of linked ordering differential equations, which just mathematically represent the regulatory relationships you see in this cartoon. I'm not going to go into the details today because we published this several years ago. But having established a mathematical representation of this circuit, we can begin to explore this behavior in simulations in silico. And one thing we notice is that this circuit very readily generates the spatial patterns of gene expression that I described to you. So in silico, if we simulate a gradient of Hedgehog signaling, so high Hedgehog signaling ventrally, low or no signaling dorsally, we see this circuit is able to recapitulate that pattern, those stripes of gene expression. So high signal generates a stripe of high response gene NKX2-2, lower signal generates a stripe of the low response gene OLIG2, and where there's very low or absent Hedgehog signaling, the dorsal genes PAX6 and IRAK3 are expressed. Moreover, these simulations were able to recapitulate the genetics I showed you. So if we remove the equation representing PAX6, what you notice is an expansion of NKX2-2 to lower levels of signal and a reduction in the OLIG2, the low response gene. And as you remember, that's experimentally what we're seeing. So in a PAX6, in the absence of PAX6 protein, we see an expansion of NKX2-2 to lower levels of signal. So this was reassuring. We had a mathematical model that recapitulated the spatial patterns of gene expression and the genetic results we'd obtained. What was more striking and more interesting to us is that the simulations also recapitulated, also explained the temporal response we were seeing in vivo. And this wasn't something that we'd asked the model to do. It's any instantiation, any version of the model that gave us the right spatial pattern of gene expression also gave us the right temporal response. So if we look at the simulation here, we're simulating with a high level of signal that at long time, at steady state, gives us the high response gene, NKX2-2, but it's always preceded by a transient expression of OLIG2. And remember, that's what we're seeing experimentally, both in explants, ex vivo, and also in vivo with this sequential induction of first the low response gene, transiently, and then the high response gene. So the same regulatory mechanism explains both the spatial pattern and the genetics of gene expression, and it also explains the temporal behavior. So we don't have to look for a novel, a distinct temporal mechanism. That temporal mechanism is encoded in the same regulatory logic that gives us the spatial pattern of gene expression. It's the same gene regulatory mechanisms that gives us temporal and spatial behavior. So one way of looking at this is by plotting an input-output relationship between the duration of signal and the level of signal. So if we look at this, so for the high response gene NKX2-2, we need a level of signal above a certain threshold, so you need high levels of signal. You also need that signal for a sufficient period of time. And to get the NKX2-2, you always transient through an OLIG2, a low response state, before achieving that high response state. And this is a consequence of that regulatory network. So you can view this gene regulatory network as a computational device, if you like, an information processing system that converts both the level of signal and the duration of signal into this appropriate pattern of gene expression. So I think one interesting thing this does is it sort of reconciles some of the controversies over whether morphogens, or whether graded signals really act as a concentration-dependent or time-dependent signal. So from the perspective of the gene regulatory network, the actual response of cells, the two are indistinguishable. It's both the same thing. You can't separate the response to the level from the response to the duration of signal. And what that also means is you can't separate position from pattern. There's no positional information separate from pattern. It's the dynamics of the gene regulatory network that generate both the positional identity and the pattern of gene expression. So in some ways, you can think of positional information as either not existing here or as emerging from the dynamics of the regulatory network. Okay, so I focused on the dorsal ventral axis and indeed a little part of the ventral neural tube. But in addition to the dorsal ventral axis, cell identity is also dependent on the other spatial axis, the anterior posterior identity of cells. So different types of neurons are formed at different rostral caudal levels. For example, you have different sets of motor neurons at limb levels compared to thoracic levels. And this also involves a gene regulatory network controlled by extracellular signals. So obviously the HOX genes, retinoic acid, FGF signaling are involved. But I'm not going to have time to talk about this today. Instead, what I want to discuss is the third dimension, if you like, is that neurons in the spinal cord also diversify along the temporal axis. And this axis depends on cohorts of transcription factors that subdivide neurons in the spinal cord independent of their position, but based on their date of birth. And this had previously been overlooked in the spinal cord. Moreover, it turns out that the same temporal program operates throughout the nervous system. And so how do we get to this? So how do we come about this realization? Well, this project started when we decided to apply, as we all are doing, the newly emerging single-cell sequencing technologies to generate a molecular atlas of gene expression in the developing spinal cord. As most neurons in mouse are born between E9.5 and E13.5, we focused on these stages and we performed single-cell sequencing using the 10x genomics platform, which I'm sure you're all familiar with, on dissected and dissociated limb level and thoracic spinal cords, taking embryos at each day between E9.5 and E13.5. So overall, we recovered about 50-odd thousand cells, of which 25,000 were neural. So here's the high-level summary of that data. So as I was saying, we had about 55,000 cells with a mean of about 2,000 genes per cell, and about half of the cells or so that we recovered were spinal cord in origin, so either spinal cord progenitors or neurons. As expected, because we were dissecting embryos, the dissections also resulted in us recovering some cells from the adjacent neural crest, mesoderm, so particularly somites, and obviously some blood and vasculature. And we could identify those based on gene expression. and we computationally eliminated those from the rest of the analysis and focused on the neural cells. So to organize the analysis, we made use of existing knowledge. So we already know a lot about gene expression in the neural tube, and we can use that knowledge to identify distinct groups of neural progenitors and postmitotic neurons. And the way we approach this is for each identities, for each cell type, either progenitor or neuron, we defined a target gene expression based on combinations of marker gene expression, and then assigned each of the cells in our data set to its closest target gene expression. So let me first illustrate this with a little summary of the assignment of progenitors to their identity. So we used a panel of progenitor expressed genes, and based on the expression pattern in each of the cells, we could identify or assign each of the cells, progenitor cells in our data set, to one of the previously defined progenitor identity. So this allowed us to classify each cell in the data set into one of the 13 previously defined progenitor domains. So for example, you can probably see here that we have a set of cells, progenitor cells here expressing OLIG2. They don't express NKX22 or NKX29, it's parallel, and those we assign to motor neuron progenitor identity. We undertook a similar exercise with neuronal markers. Again, here's a panel of neuronal expressed genes which we could use to assign each of the neuronal cells in our data set. And again, we could classify each of the neurons into one of the 13 cardinal classes of neurons. So now we have a data set where we've classified about 25,000 cells into approximately 26 subtypes, 13 progenitors, 13 neuronal classes. So the first thing we wanted to do is some validation. So is our assignment, is this analysis, does it make sense? So one thing we can do is, since we know the age of the embryo from which each cell was recovered, we can use this to predict changes in cell type over time. So as you might expect, over this time course, the proportion of progenitors goes down over time as the proportions of neurons increases. In addition, we can see changes in the relative proportions of these different cell types over developmental time. And we can compare these inferred changes in cell type identity to previous measurements we collected. So independent of this, we had collected a data set where we looked at the changing proportions of neurons and progenitors over time, and we can compare that to the inference from our single cell transcriptome data. And reassuringly, these two data sets gave us a reasonable match. So that suggests that the dissociation library preparation and the analysis wasn't systematically biasing the cell types we were recovering. It's not that we were losing neurons or losing progenitors disproportionately. So our classification scheme was producing an output which was consistent with an independent assay. A further test of the accuracy of the data, we could use neurotransmitter and transporter expression to predict the physiological properties of each of the neuronal classes we'd assigned. And this allows us to split the neurons into excitatory and inhibitory interneurons and cholinergic motor neurons. And again, these predictions matched our prior knowledge of the assignment of excitatory and inhibitory interneurons in the spinal cord. So taken together, the data aligned well with our previous knowledge and suggested that this atlas, the transcriptomes we recovered, was giving us a good representation of the developing spinal cord. So we then wanted to determine the resolution of the data set. How fine-grained could we assign subtype identity within the data? So previous work has revealed that each of the cardinal neuronal classes can be further partitioned into further specific subtypes. So here I'm showing you the example of V2A interneurons, all of which express the transcription factor CHOX10. which is also known as VSX2. But this V2A class of neurons can be further divided into subtypes based on the expression of further transcription factors. So, for example, Harris et al. Had defined a subgroup expressing the one-cut family of transcription factors, which is distinct from a subset expressing SHOX2. So, could we see this in our data sets? And reassuringly, yes, we could. So when we isolated the V2A transcriptomes from our data, identified based on expression of CHOX10, and then recluster those, we saw that a subset of those neurons expressed SHOX2, and this was distinct from a subset which expressed the one-cut family of transcription factors. Moreover, what we saw is, in addition, a further subset which expressed genes, a gene module including NF1A, NF1B, NeuroD2, which hadn't previously been described. And indeed, when we looked for these, when we went back in vivo, we could indeed see a subset of V2A neurons, which indeed expressed NF1B, NeuroD2. So that was encouraging. We could recover from the transcriptomes what was already known about subtypes of neuronal classes, and indeed identified previously undescribed subclasses. So that encouraged us to go ahead and to further partition each of the cardinal neuronal classes within our data set. And when we did this, we found each of those cardinal subtypes could be split into multiple subclasses. And what was really striking is that the same transcriptional signatures, the same gene modules, were distinguishing each of these subclasses. So for each interneuron subclass, we saw, for each cardinal neuronal class, we saw a subtype which expressed one cut, another subgroup which expressed a gene module including PAL2F2, ZFHX3, and a third partition, third gene module which expressed NeuroD2, NF1A, NF1B. And remember, these are spatially, molecularly, and physiologically distinct neuronal classes, but they're being further divided by a recurring transcriptional code that subdivides each of them. So what explains this? So as I mentioned, we know the embryo age of each of the cells in our data set. And when we examine this, we realize that the same transcriptional code correlated with developmental age. So the first neurons born from each of the progenitor domains, irrespective of their dorsal ventral identity, expressed the one-cut gene module. Then about a day later, neurons from each of the domains were expressing that PAL2F2, ZFHX3 module. And then a day or two further later, that the neurons generated from each of the domains was expressing NeuroD2, NF1A, NF1B. So this suggests a temporal program stratifies neurons independent of their spatial identity. So this provides an additional access to for neuronal diversity. So in addition to the molecular code that distinguishes neurons along the dorsal ventral and anterior posterior axis, there's an orthogonal molecular code that distinguishes the temporal identity of neurons. Okay, so to test that idea directly, we wanted to birth date neurons, and to do that we use sort of the well-established EDU birth dating approach. So this technique relies on EDU incorporation into proliferating progenitors. Obviously, postmitotic neurons don't incorporate it. And since they don't incorporate it, then the levels of EDU in the nucleus allows us to determine when neurons were born relative to the time when we've injected EDU. And consistent with that transcriptome data, EDU injection at 9.5 labels ZFHX3 neurons, but not NF1A, NF1B, NeuroD2 neurons, whereas EDU injection at a later time labels that later gene expression module, so the NeuroD2, NF1B expressing neurons, but not the earlier generated neurons, the ZFH3 module, the early born subtypes. So confirming that temporal sequence that we saw from the transcriptome data. So taken together, these data suggest a temporal program in the spinal cord operating orthogonally to spatial patterning that increases the neuronal subtype diversity. But is this unique to the spinal cord? So to explore this, we took advantage of transcriptome data generated from other regions of the mouse central nervous system. So here I'm showing you some data from Sten Lennerson and colleagues who generated single-cell transcriptome data from forebrain, midbrain, and hindbrain. And strikingly, when we analyzed that, we saw the same temporal sequence. So the first generated neurons in each case were expressing one cut, then you transitioned into a phase of generating ZFH3 expressing neurons, and finally at later stages, this NFI NeuroD2 stage, this NeuroD2 temporal program. I should say we also found the same sequence in the retina. I'm not showing you that detail here. It looks like the entire nervous system has this same temporal code elaborated during this sort of phase of E9.5 to 13, 14. So what are the consequences of this? So to give you a sense of this, I just want to highlight the dopaminergic neurons in the midbrain. So previous characterization of these neurons by Thomas Perman and colleagues, where they generated single-cell data using PIDX3 transgenic reporter, suggested that dopaminergic neurons can be subdivided into several subtypes, including a ZFX3 subtype and an NFIA subtype. And in fact, what Perman and colleagues concluded is that it's a ZFHX3 positive population that is the bona fide dopaminergic neurons. They express the appropriate dopaminergic transporter and high levels of the rate-limiting enzyme TH. So consistent with this, when we looked in vivo, we found that ZFHX3 neurons generated at this intermediate stage at 11.5 in the midbrain. They expressed the classic sort of dopaminergic marker NMX1B, also TH, the synthetic enzyme. Whereas the later born NF1B neurons are not present as neurons at 11.5, and then by 13.5, you can begin to see NF1B neurons, but they're not expressing the same set of markers. So dopaminergic neurons correspond to this intermediate time point, this ZFHX3 positive population generated in this middle time window of this temporal code. Moreover, the temporal program is not limited to in vivo derived neurons. We see the same temporal sequence of neurogenesis progressing at a very similar rate from in vitro differentiated neurons. So using established protocols to differentiate embryonic stem cells into either dorsal or ventral spinal cord neurons, we can see that the first generated neurons, as in vivo, express one cut. Couple of days later, we see the POW2F2 ZFHX3 neurons, and then finally 2 or 3 days later, still we see that NeuroD temporal program kick in. I should say we see the same sequence of from in vitro neuron differentiated from ES cells to either midbrain or hindbrain identity. So that's the temporal program in neurons. I mean, I think what it suggests is that this temporal program is dependent on some kind of cell intrinsic or local signaling. It argues against a global pacemaking mechanism outside of the neural tube, or indeed it suggests that you don't need secreted ligands from specific signaling centers such as roof plate or floor plate, since these are not present in these in vitro differentiations. So this prompts a question of what signals, what gene regulatory mechanisms might underlie this temporal patterning of neurons. So previously, both Johan Eriksson's lab, Lorenz Studer's lab have implicated TGF-beta signaling in the transition of neural progenitors to the generation of glial cells. So we wondered whether this could also influence the broader temporal program of neurogenesis. So consistent with that idea, we found that inhibiting TGF-beta signaling using a small molecule inhibitor in vitro delays the progression of the temporal program. We see delays in the acquisition of the late gene module expression, so NF1B, so NF1A here, and NeuroD2 expression is delayed by a day or two in the presence of the TGF-beta inhibitor. And conversely, if we add recombinant TGF-beta, we see premature progression of this program. However, I think it's important to note and emphasize that progression still occurs in this case. Inhibiting TGF-beta doesn't abrogate progression, it just delays the switch. So it's not solely dependent on TGF-beta. There must be something else. We don't yet know what it is, but it suggests again that there must be some, or raises the possibility of some cell-intrinsic mechanism involved. So if we think about those cell-intrinsic mechanisms, I think it's fair to say that temporal generation of different types of neurons from the same progenitors is arguably best understood in Drosophila. So in Drosophila, Christou, Claude Desplan, and their colleagues have found that aging neuroblasts sequentially express a series of transcription factors that define temporal identity windows, resulting in the generation of specific cell type progeny. Similar processes are believed to underlie temporal patterning in the vertebrate nervous system. However, I think it's fair to say that temporal programs, transcriptional programs that mediate this are relatively poorly defined. So to tackle this, we returned to our single-cell transcriptome data and now interrogated gene expression in spinal cord progenitors. And this revealed what looks like a spatial dorsoventral independent temporal program also operating in progenitors as well as neurons. So this includes some familiar friends being implicated in temporal control of patterning in other systems, including LIN28A, SOX9, NF1A, NF1B. And what's striking is the timing of the transitions in the progenitors coincided with the temporal transitions or temporal switches we're seeing in post-mitotic neuronal identity. Moreover, we see this is not only in the spinal cord, but also in other regions of the nervous system as well. So is any of this relevant to the temporal identity of post-mitotic neurons? So we've begun to investigate that. So we've started to look at the function of specific components of the progenitor program. And we've used CRISPR to eliminate here NF1A and NF1B from ES cells. And while these ES cells remain competent to differentiate into the intermediate temporal subtype, exemplified by ZFHX3 expression, the absence of NFI activity results in a marked reduction in that late subtype, the NeuroD subtype of neurons. And we're obviously keen to continue investigating this to better understand how the progenitor GRN, the regulatory network in progenitors, affects the temporal identity of post-mitotic neurons. So let me summarize these data. So what I tried to show you is that single-cell transcriptome analysis revealed a previously sort of unappreciated temporal program of neurogenesis in the spinal cord, which we could confirm directly by EDU birth dating. There seems to be a similar program operating throughout the central nervous system, including midbrain, forebrain, and the retina. And this suggests this provides a common central nervous system-wide mechanism that increases the diversity of neuronal subtypes, operating in parallel to perhaps a much better understood spatial patterning processes. I also showed you evidence that the program operates in vitro, suggesting the progression is driven by cell-intrinsic timers or at least local signaling. And finally, we have a little bit of evidence that TGF-beta signaling influences the speed of this temporal program, most likely by influencing gene expression in the progenitors. And I think that's particularly intriguing because TGF-beta signaling has also been implicated in controlling the timing of fate switches in Drosophila mushroom body. So that raises a possibility of a deep evolutionary conserved role for TGF-beta signaling in controlling temporal patterning of the nervous system, and suggests this might be a mechanism to increase neuronal diversity throughout the bilaterian clade. Okay, so I want to briefly finish in the last 5-10 minutes or so by talking about another aspect of development that's encoded in time, and that's the pace of developmental processes. So obviously the pace of embryonic development determines the overall duration of gestation and also the rate of individual developmental processes. And I think what's striking is that despite the similarities in developmental progression between different species, particularly if we think about different mammalian species, the time that this takes is very different. It's really strikingly different between different species. And you can see here an example of comparing on the top mouse embryo development to human embryo development on the bottom. Whereas mouse embryo development takes about 20 days, the equivalent period of human development takes 9 weeks, 60 days. In humans, this is then followed by the next 31 weeks of fetal stages of development. But we were interested in sort of understanding this early period, and we wondered what are the mechanisms behind this difference in pace of these early developmental stages. I think what's particularly striking is if we look at embryos, we can see, it's very difficult to distinguish them. So here I placed a mouse and a human embryo next to each other at equivalent stages of development, and there's a little test for you here. So you've got 10 seconds to see if you can tell which is the human embryo and which is the mouse embryo. So I hope you see there's an obvious clue in the image that I made it easy for you. So here's the mouse tail, and you can see the human tail is much smaller. But I hope it's obvious. So, I mean, particularly with the heads removed from these two embryos, it's very difficult to distinguish which is a mouse and which is a human embryo unless you have some prior knowledge. Moreover, you can see that these embryos at these stages are very similar in size. And this similarity is also evident if we compare in detail the patterning of patterning process. So here, the patterning of neural progenitors, comparing the mouse neural tube to the human neural tube. And I should say these human embryos were collected from elective terminations through the Human Developmental Biology resource in the UK. So we can see by comparing the mouse and human embryos, patterning of progenitors is highly conserved. We're seeing the same stripes of progenitors in the same relative positions, and also the same relative evolution of these patterns over this developmental staging series. Perhaps as you would anticipate, this is a highly evolutionary conserved process, spinal cord patterning. But despite this conservation of the spatial pattern, what does differ substantially between the two species is the timing. So mouse sections I showed you covered embryonic day 9 to embryonic day 12, so about a 3-day period. And by contrast, the human sections were from Carnegie stage 11 to Carnegie stage 17. So that's a period of more than a week, so actually about 2 weeks. So this highlights that the development of the neural tube and the elaboration of that gene regulating network, despite the similarity, it's happening at a very different pace in the two species. And I think this gives us a specific, a well-characterized system in which we can explore where these differences in tempo arise. And one way we've started to tackle this is to, again, turn to in vitro systems. So as I've shown you, we've developed the directed differentiation of both mouse and human embryonic stem cells into spinal cord progenitors, and specifically to the differentiation of motor neurons. For the sake of time, I'm not going to bore you with all of the details of these, but just to emphasize that the differentiation protocols mimic in vivo developmental events. They use the same signals, the same conditions, and they produce the same types of neurons following the same sequence of gene expression in both species. But despite that similarity, in vitro as in vivo, these two differentiation processes unfold at very different rates. And that's perhaps easiest to see if we compare the dynamics of specific gene expression. So if we look at the key transcription factors in the generation of motor neurons, we can see that initially neural progenitors express PAX6. They then turn off PAX6 as they turn on OLIG2. And again, they begin to turn off OLIG2 as they either differentiate into islet expression motor neurons or into P3, so another progenitor domain expressing NKX2-2. So that same sequence of events was also seen in human ESL differentiation, but the pace of those transitions was substantially slower. And comparing the speed of differentiation between the two species, we can measure a fold difference in the time it takes, and time for the appearance of PAX6, OLIG2, and NKX2-2. And for each of these genes, that factor, that temporal scaling factor, is approximately 2.5. It's between 2 and 3. So humans are doing the same thing, but about 2 to 3 times slower than the mouse. This is also true if we analyze the whole transcriptome. We see the same phases of gene expression in the same sequence, but they're happening at a different pace. And when we measure this difference, we again find a temporal scaling factor about 2.5-fold, and that appears uniform across this whole developmental period. So I think this says a couple of things. So first of all, it suggests that tempo is an intrinsic property of cells. Again, it's not dependent on signals external to the spinal cord. We're seeing it in vitro in differentiating in vitro cells. Moreover, this uniform scaling of tempo suggests that the difference doesn't arise from a particular bottleneck in humans. It's not a consequence of development running at exactly the same speed to a specific point and then stopping. But it's more likely a consequence of proportional scaling throughout the whole differentiation process. So what could explain this difference in tempo? Well, it doesn't seem to be differential sensitivity to signals. We can again test this in vitro. If we look at the response of human and mouse cells to exogenous added recombinant hedgehog protein, we don't see any difference. Here I'm illustrating this by comparing the kinetics of activation of a couple of well-documented sonic responsive genes. They respond similarly in human and mouse neural progenitors. So an alternative, another possibility would be the difference in tempo could come from differences in gene regulation. You can imagine there's cis-regulatory differences between mouse and human, with human being less sensitive. So in human, everything takes longer because it's less sensitive. So to test this, we've taken advantage of a mouse ES cell line that contains the human chromosome 21. These were made because obviously human chromosome 21 is involved in Down syndrome. But it happens that human OLIG2 is actually situated on human chromosome 22. So we have our favorite gene just happens to be on human chromosome 21. So now we've got a human OLIG2 on its human chromosome, but in a mouse cell. And we can distinguish between mouse OLIG2 transcripts and human OLIG2 transcripts using single molecule FISH. So as you may remember, mouse OLIG2 induces day 2 in our differentiation protocol, and that's true in the human chromosome 21 containing mouse cells as well as in control cells. From the previous results, we know that human OLIG2 in human cells takes 4 to 6 days to be induced. However, the human OLIG2 in mouse cells takes on the mouse timing, and it's expressed by day 2 of the differentiation. So human OLIG2 in its human genomic context is induced more rapidly in mouse cells than in human cells. So it suggests that the local genomic sequence, the local cis-regulatory elements, aren't determining the tempo of gene expression. So it doesn't seem to be differences in gene regulation either that's responsible for this tempo. So what are we left with? So another possibility is that some other kinetic features of gene expression that explain this difference in tempo. And we designed a series of experiments based on pulse chase labeling to measure both RNA and protein stability. Won't go into the details, but RNA stability seems to be similar in mouse and human cells. So overall, the transcriptome has a half-life that averages about 90 minutes in both mouse and human neural progenitors. By contrast, however, if we measure the proteome in human and mouse, we find the human proteome is more stable. So when we estimate a half-life, we find the proteome in mouse neural progenitors of a half-life of around about 8-9 hours. By contrast, in human neural progenitors, the proteome has an average half-life of around 18 to 20 hours. It's not only average proteome, the proteome as a whole, but if we look at individual proteins, we also see this 2- to 3-fold difference in stability. So here, looking at a couple of favorite transcription factors, you can see for NKX 6.1 and for OLIG2, this 2-fold difference between mouse and human. So OLIG2 protein has a half-life of 4 hours in mouse cells and about 8 hours in human cells. Okay, so there's this 2-fold difference in stability of proteins correlating with this 2-fold difference in tempo. But of course, protein turnover controls several cellular processes, perhaps most notably cell cycle progression. But conversely, cell division also dilutes protein, so changing the cell cycle rate alters the rate of protein turnover. So what's the chicken and egg here? So this prompted us to ask whether cell cycle speed was also a factor in our observations of protein half-life. And the first thing we did was measure the proliferation rate of mouse and human neural progenitors, here using cumulative EDU incorporation. And this revealed that the cell cycle length was approximately 2.5-fold longer in human cells, in human neural progenitors compared to mouse neural progenitor, again fitting that general 2.5-fold temporal scaling. But this raises a question then of whether this difference in the cell cycle is contributing to this difference in protein stability. So to address this, we've measured protein stability in mouse and human motor neurons. So these are obviously post-mitotic non-cycling cells. We isolated motor neurons in vitro by flow cytometry, again using the same pulse chase method we established in neural progenitors to measure protein half-life. And this revealed that similar to proliferating cells, the proteome is more stable in human motor neurons than in mouse motor neurons. And again, the central estimate for this difference in half-life suggests a 2- to 3-fold difference in stability. And I should say the confidence intervals for each species overlaps with the same species from the progenitor experiments. So this suggests that this difference in protein stability between mouse and human is not simply explained by differences in cell cycle length. And I'd actually argue that the causative relationship is more likely to be the other way around, that the more stable human proteome is retarding cell cycle progression in human cells. So the question is then, so can these changes in protein stability account for differences in developmental tempo? So to test this, we used the computational models I'd introduced to you earlier of the gene regulatory network that recapitulate in silico the progression of gene expression during neural development. We can simulate these models now with the values for protein turnover measured from the experimental data. And if we do that for the mouse experimental measurements, this gives us a characteristic timescale for the dynamics of the gene regulatory network. So having identified a 2-fold difference in the stability of human proteins, we can decrease the degradation rate 2-fold in the simulations and ask what happens. And what you can see here is that this increased stability of proteins in the simulations results in the slowing by a factor of about 2.5-fold in the rate of progression of the gene regulatory network, suggesting that changes in protein stability are sufficient to account for the difference in developmental tempo. So let me wrap up here. So in summary, I've argued that protein stability plays an important role in controlling developmental tempo. The mouse motor neuron developmental program progresses twice as fast as the human. And this, at least in part, appears to be a consequence of that twofold difference in protein degradation. So human proteome is more stable and the development is slower. We're, of course, interested in directly testing this hypothesis and understanding what determines the global rate of protein turnover. And I'd also like to highlight that Miki Ebasuya's lab have nicely complementary findings looking at oscillations of the mesodermal segmentation clock, comparing mouse and human. So I think together these results indicate that proteostasis has an important role in setting the tempo of development. And we'd hypothesize that if we could modulate protein stability, we could modulate the speed of differentiation. So at the end, I want to come back to that question I posed at the beginning. So how are hundreds of diverse cell types generated in the neural tube? And what I've argued is that intersecting temporal and spatial programs of gene expression, which are closely choreographed, result in these intersecting orthogonal axes producing combinatorial complexity to generate the diversity necessary to in that first step in the assembly of functional neuronal circuits. And I think the challenge now is to understand how these operate and what explains features such as the remarkable precision and robustness of the process. So finally, I'd like to thank members of the lab. I've highlighted in particular the work the people involved in the work I discussed today. The tempo work was spearheaded by Teresa Rayon, Despina Stamataki, Ruben Perez, and the temporal program throughout the nervous system, spearheaded by Andreas Sagner with Isabel Zhang and Julian DeLille. And thank you to our colleagues, Chris Barrington, to Victor Tabulowitz and Lizzie Fisher for their collaboration. And thank you for your attention.
Thank you very much, James. It's this awkward moment where nobody's clapping, but if you look, I can actually physically clap. It's even more awkward when I do that. But that was great. Thanks for driving us through this beautiful array of data. People are not clapping on the right there if you want to have a look. So there's we have time for a few questions, and even for more than a few questions. So I'll read them out to you, and we can start the discussion. So the first one is from Shubha Tole. Hi, James, enjoyed your talk. You mentioned AP patterning in the first part of your talk. How do you think this is regulated in animals that are vastly different in length? The embryos start out not very different, so the RA gradients may not be that different, yet different segments of the spinal cord with their distinct neuronal compositions need to be very different in length depending on the animal.
Yeah, I think that's a great question. I think obviously you should pose that to people like Olivier Porquier and Miki Ebasuya. But I think a striking comparison, if you like, is that AP axis, the laying down the posterior axis of the neural tube, also depends on the temporal program. So Hox genes progress, as you know very well in Geneva with Denis Debour's work, progress through a series of increasingly more posterior identity, and the rate at which that progression happens defines the AP identity of the segments being generated. So again, there's a relationship between the spatial and temporal patterning here, which again, I don't think anyone will claim to fully understand, but somewhere in there must be an explanation of how you generate the appropriate sized segments, regions, rostral caudal regions of the forming spinal cord. And I think it's striking that, again, it's an interaction between time and space to do that.
Mm-hmm. Thank you. Next question is from Tim Monco. Thank you for the talk, James. I'm wondering how human cell cycle length is longer, while human progenitors seem to have a greater proliferative capacity. If cells are dividing less, then if cells are dividing less, then less cells are being born. I believe that in mouse, it's been identified that cell cycle lengthens as progenitors become more neurogenic rather than proliferative. How do you think cell cycle length relates to this?
Yeah, great question.
Increased protein stability related to this effect.
Yeah, I mean, I think the short answer is we don't know. You're absolutely right. And I oversimplified things. Of course, the cell cycle actually increases in length over this period of time. So you actually have, on top of the difference between species, you also have a change over time in the cell cycle length. What controls that is unclear. Of course, we know, broadly speaking, that protein degradation is important for the progression of the cell cycle, so degradation of cyclins. So it seems quite likely to me that proteostasis, so protein degradation, will contribute to this. But the details, I think, are unknown. And I find it actually fascinating to think about these questions, because overall you have the species differences, and then you have a difference in developmental time as well.
But the slope of the increase in the cell cycle length is the same in mouse versus human?
So we haven't measured that in vitro. In vivo, of course, it's going to be difficult or impossible to get, because you'd have to infer that from static or very short-term culture of human embryos. So I think getting it from human embryos would be difficult. I think it would be easier to do if comparing other model species. I'm not, off the top of my head, I'm not aware that it's been done in the neural tube, but it is an interesting question.
But you looked at different embryonic ages, right? And you were saying it's constant, so that would argue that the slope is the same, or not?
So the difference is constant. So we're seeing within the limits, the areas of our experiments, we're seeing a twofold difference in the cell cycle, but they're both changing over time.
Mm-hmm. And so how do you reconcile this with, I don't know how that is in the spinal cord, but in the forebrain, the greater number of neurons being generated?
Right, and I think that comes down to then the interaction between cell cycle rate and the rate of differentiation. And I think an interesting, so one reason, I'm going to focus on the spinal cord because I love the spinal cord, but an interesting or an important aspect of the spinal cord is that it's very evolutionary conserved. Whereas the cortex in particular, there have been selective pressures for changes in size. So there may be derived features in the cortex about changing the rate of differentiation or the cell cycle, which is specific to the cortex, which you don't find in other regions of the nervous system. I'm speculating, but that could be the case. I think it's an interesting question to ask is whether the difference, how much of the difference in the size of primate cortex compared to rodent cortex can be explained by the mechanisms that control the difference in the rate of development of the spinal cord, or have additional mechanisms also being incorporated to explain that difference in cortical size.
But if you were the one that were designing the rate of cell division, right, I don't think you would choose a half-life of proteins as the way to intervene there as the driver, or would you? I mean, it seems dangerously downstream, and evolutionary pressures there.
Yeah, and I think that also raises other interesting questions, because if you change just the degradation rate, that has also knock-on consequences. So if you only change that and nothing else, then that will have an effect on molecule number comparing to, and that will obviously have an effect on things such as noise, so stochasticity. I think we need to do much more work to really understand the consequences of this. So are we seeing a major difference in protein number in human versus mouse cells? I don't think so, but we need to do more work on that. If not, that implies there's other compensatory changes as well. Then what controls the rate of protein degradation? Is a master regulator? I'd speculate metabolism is likely to be involved. is this actually an effect of change in overall metabolic rate between mouse and human? If so, how is that connected to rate of protein degradation? There's a lot of questions there, and I mean, it's very speculative. There's a lot of unknowns.
Is there any work on prokaryotes on this?
So in terms of protein turnover, yes. I mean, an important difference to mention in prokaryotes, of course, or at least a lot of the work that's been done in prokaryotes, is that cell division is the major mechanism of protein turnover in prokaryotes, because typically when you grow them in the lab, you've got a cell cycle time of 20 minutes, 30 minutes, whereas,
But I meant in terms of duration of the cell cycle relating to protein stability.
So I mean, you do need protein turnover. I would have to reach back to my microbiology background 25 years ago to remember details of DNA replication and cell division in prokaryotes.
Well, if there's anyone in the audience that doesn't have to go back 25 years, let us know. All right, a question by Sergi Roy Prigros. Does this spinal cord development tempo between mouse and human, so the difference, correspond to the ones observed in cortical development? Do you think that the same mechanisms ensuring these features are conserved across developing CNS regions?
So I've already tried to answer that, and the short answer is don't know. I think it's a very interesting question. So those differences in cortical development, have they exploited the same mechanism or used different mechanisms?
What about non-coding RNAs, and in particular microRNAs, which are known to increase in more evolved species in controlling mRNA stability? So you showed it doesn't seem to be directly involved, but are they out of the game fully?
We haven't. I think that's a good point. It's something we could test. Though, for example, we haven't looked specifically at DICER mutants. So is there any difference in tempo? Yeah. If you notice, in the middle part of my talk, looking at the single cell transcriptome data in the temporal program, Lin28A, which obviously homologue of the C. elegans heterochronic pathway, component of the heterochronic pathway, is implicated. So, I mean, don't know, but an interesting question.
Okay. Pablo M., great talk. Do you think that the slow tempo is caused by a modulation of the ubiquitin proteasome system? Any speculation of the intrinsic signaling that can cause this difference in mouse and human mechanisms?
I mean, I can speculate. As I said, my speculation, at least this month, is metabolism. So something about the different metabolic rate. The hypothesis would be differences in metabolic rate between mouse and human are controlling a range of processes, including protein degradation or proteostasis more generally. The details of that mechanism, I have no idea.
That one is from me, actually. I wrote it down while you were talking. I might have missed it, but what is the half-life of olig-2 protein in the H21 mouse cells? Is it human or mouse-like? If you look at the protein level in this very...
Yeah, so the problem is we're just in... So the antibody recognizes mouse and human protein equally, so we can't distinguish that with the antibody. So we're in the process of making the cell line so we can distinguish the mouse and human protein.
So you want to make a bet?
I bet it will be the mouse protein.
Okay, so it's recorded. Last question. It's not a sponsored question. It refers to my lab's work. Fernando Garcia Moreno. Denis published that the competency of cortical progenitors to generate different types of neurons changes over time due to a change in their membrane potentials. Could this be related to the temporal gene network that you just described? One cut, POU2F2, NFIB. So basically, I guess the question is more anything outside of the genes themselves that may be driving the progression in identity.
It's a good possibility. I think, Denis, you can answer this. I think your data on this is later, is looking at slightly later times than we're looking at. I think
it's at later times, and you could argue that I was going to say that the electrophysiological properties and diversities of the cells is bigger, but that's probably wrong. So I just don't know, or maybe the input dependent, but even that might be wrong. So I'll answer, I don't know. That's a good answer, I think, today.
Yeah.
Which just...
There's more experiments to be done.
All right, cool, James. It was a pleasure to have you. Thanks for sharing this wonderful work.
Thank you.
Next week, the host will be Laurent Nguyen, and the guest will be Valérie Castellani from Lyon. So stay tuned for more, or actually see you then, or see them then. Have a good week till then. Bye-bye, everyone. Bye, James.
Bye.